All right. I guess I'll get into it. Hello, everybody. My name's Young. I'm studying psychology and biology with a specialization in behavioral biology. And this is an adapted version of my honors thesis for the Psychological and Brain Sciences Department, titled Development and Assessment of a Manual Tracing Algorithm for Dense Reconstruction of Neurons Labeled Using Transgenic Zebra Finches. So what are we trying to study? Broadly, we're trying to study adult neurogenesis. And adult neurogenesis is the birth of new neurons in the adult brain. And there's a lot of interest in studying this because of its potential therapeutic value in treating neurodegenerative disorders. Neurodegenerative disorders involve brain tissue dying, and if we can figure out a way to regenerate it, that might be a potential treatment. Now, the animal model we use to study this is the songbird, more specifically the zebra finch. And you might be wondering, this is quite a niche population, maybe why are you using birds instead of mice, right? And the reason for this is because adult neurogenesis is a phenomenon that was actually first convincingly identified in zebra finches. And there's another reason for this as well, is that adult neurogenesis happens in a very limited capacity in the mammalian brain. And so that's also another reason for studying it, is you want to figure out why it doesn't happen more often in the mammalian brain. And specifically what we're interested in studying is this process of neuronal migration. In adult neurogenesis, cells are born, they migrate across the brain into their site of integration, and then they integrate and differentiate. Okay, cool. Adult neurogenesis is really cool. What are we going to use to study it? Well, the technique we're using is called dense reconstruction. And what dense reconstruction entails is densely labeling a bunch of neurons in a population. And then using that, we can image that and then study the coordinated migration. Because neurons are densely labeled, you do have a lot of advantages. One, you generate a lot more data. Two, you can study interactions between neurons. And three, you can study this coordination or population level dynamics. Now, this is really cool, but there is a bit of a bottleneck in this process. Before, it used to be that we couldn't image and label so many neurons. But now that we can, the trouble we have is turning these images into actual quantitative data. Now, historically, studies using cell tracing have been carried out by individual, highly trained experts. And the justification for this is because they're experts, they have much better judgment, and you can probably rely on their data. The issue with this is because we generate so much data with dense reconstruction, though, it takes too much time for an expert to go through all that data. And so what alternatives do we have? One, we could use AI, machine learning, automated methods. And these have proven to be quite successful, but even these require a substantial amount of ground truth data in order to train algorithms. So it's unavoidable that you have to do some manual analysis. Another option is using a crowdsourcing approach. Well, individual experts might be unfeasible. Maybe you can take a bunch of less experienced people, pull their data together, and see if that generates data similar to an expert. So here is some of the data we generated, and I'm going to go through some of my methods at this point. So we generated this data using transgenic zebra finches. These are zebra finches that have been bred to basically have fluorescent neurons. They glow. And what we can do with this is we can stick a camera in there and generate a volumetric time lapse of their brains. And this basically just means a 3D movie of their brains. And you can see here in this image that we have neurons that are migrating, which is very exciting. And this is specifically the population that we're interested in studying. Now, these images are cool, but there's not much you can do with just images. We have to figure out a way to get quantitative data out of these images. And that is done through tracking. And what tracking entails is finding the center point of every single neuron at every single time point your data has. And this allows you to reconstruct their trajectories and answer biological questions with that. Now, some of the goals we want to get through. First, we want to examine manual tracking as a methodology. We want to see how it works in both experts and novices. Two, we want to see whether we can pool together novice data in order to get expert-level data. This lends credibility to a crowdsourcing approach. And finally, we want to use this method to actually generate a data set of neuronal trajectories, which we can use to answer questions about neuronal migration. And so this is kind of a visual understanding of the project. You have your migrating neuron on the left, and basically what we want to do is we want to track this neuron, find its coordinates across all time points, and generate a reconstructed trajectory of it. So let's go through some of my results. The first result we found is that between individual experts, there's a lot of variability between their tracks. Now, like I said before, historically, people have relied on the individual expert level tracks in order to study these things. But we found that even between experts, there's only about a 50% crossover between their tracks. And what this means is that we can't really rely on just individual experts in order to generate ground truth data. What we can do instead is maybe use the consensus between experts to generate data. This means taking multiple experts, having them all track the same thing, and then only using the intersection of their data. That will generate a much more reliable set of ground truth data than just individual experts. What you can see here is also that novices are much worse than experts in terms of their variability as well. And what this means is that we'll have to pull together even more novices if we want to reach expert level. Our second finding is that individual novices are less accurate than expert. When I say accurate, this means that they basically don't catch as many ground truth neurons as experts. And this is something we expected. Obviously, the people we recruited are only recruited for this purpose. They have no prior knowledge and they have no experience. And so it's expected that they would be a little worse at it than experts. But we found two ways that we can actually improve this accuracy. The first way you can improve it is implementing a training model that incorporates real-time feedback. When I recruited trackers for this project, I trained them using either this kind of model, which incorporates real-time feedback. I work with them in person. Or I send them training materials and have them just follow a video and documents. And what we found is that the trackers who were trained using just the documents performed much worse than those who got feedback. And I think this makes sense as the real-time feedback part is really mimicking what an expert would go through. An expert would likely go through multiple rounds of training, would likely be trained by someone else who is expert as well. And so this makes sense to me. And finally, our last finding is that while individual novices are not as good, you can take the union and pull together 3 novices' data and generate data with accuracy similar to an expert. And this is really the finding that we wanted to find, because this lends credence to this possibility of using a crowdsourcing approach to generate data. So what do I want to do with this? Well, our first step is to assess other factors that affect tracking. Other projects who have used crowdsourcing approaches to generate dense reconstruction data have looked at different factors like motivation. Intrinsic and extrinsic motivation are certainly very important, and it's also an aspect of why experts perform better, I think. Experts tend to be much more involved and motivated than novices. Novices might see it as just another task to do, while experts have their careers maybe relying on the data. Because we've seen that we can pull together data and generate expert-level accuracy, we want to maybe scale this up a bit more. One thing that this would involve is developing some sort of crowdsourcing platform. Right now, I'm only just personally communicating with trackers, and it's a bit tedious on my part, but we can develop a better platform, maybe having individualized profiles for novice trackers. We can better generate crowdsourcing data. And then finally, we want to use the data we generate to actually answer biological questions. In some comparisons to previous literature, we found that the data we generate is quite similar to existing data, which again lends credence that we can use this methodology to generate usable data. And so the next step is actually to use this data to answer questions, which my grad student Naomi is actually working on right now, excitingly. And that is the end of my presentation. I'd like to give a couple of thanks here. First to my PI, Dr. Ben Scott, who has guided me throughout this entire process, Dr. Steve Ramirez and Dr. Joe McGuire, who are members of my thesis defense committee, Naomi, who's my grad student mentor and has been invaluable in guiding me through this process, members of the Scott Lab, which I performed my research in, and finally the Department of Psychological and Brain Sciences, for which I did the honors project through. All right, any questions?
Hi, I was wondering if you could share a little bit about the time span of the neuronal migration. Is it happening on the order of like seconds or days, or what's the time span for like that video we saw?
Yeah, so let me go back to the video. This entire video is from a 3-hour imaging session, sped up to, well, I played this at about a frame rate, about 20 frames per second. And each time frame was captured at about a 10-minute interval. So this is the migration you would expect over a 3-hour period.
Can I ask you a question going back? So I know the bulk of your presentation is on the methodological intervention you made in crowdsourcing. kind of any kind of data in this, but I want to go back to the specificity of the neurogenesis and zebra finches in particular. So I wonder whether you've thought about or read about the ways in which songbirds in particular are have been identified as great sites for looking at adult neurogenesis, and whether that has something to do with song, and whether you have thought about linking this to some of the research on the relationship between music, and particularly song, and memory, and maybe how that then translates to think about this in the adult brain, like should we be singing more?
Interesting. Well, you were right in that a lot of the adult neurogenesis is related to songs. And that's another reason for choosing this model, is because that adult neurogenesis you see is very tied to behavioral results. In fact, and specifically for zebra finches, most of the adult neurogenesis you see is in an area called the high vocal center. And you'll see a burst of adult neurogenesis in this area prior to song learning or during song learning for male zebra finches. In terms of translating this to a human model, I actually don't know of much literature and personally haven't looked into it much, but I think that's quite an interesting topic to study. I don't know if there's an auditory circuit that might be related to it in human brains, and that's something definitely to look into.
I'm curious about the motivations of the novice trackers you were working with. You mentioned that studying motivation further might help improve their results. Do you have guesses now about what would motivate people to volunteer to do this sort of work?
Yeah, I can tell you right now that the undergrads I recruited to do this are motivated by having research experience in a lab. And that's kind of the primary population we drew from. There's a lot of undergrads who reach out to the lab for research experience, and the first thing I did was just recruit them into this in order to get them a little bit of research to get started, as well as benefit my own research. There have been studies in the past who have scaled this up to like a very large gamified platform. And so one option is actually to gamify this task, make it actually more interesting intrinsically. Another option is that, or another possibility is that people are motivated by generating scientific data and motivated by an interest in science. And certainly that's something that the other study who did this in a crowdsourcing platform found. You can ask me later. All right. Thank you, everyone.